Imagine a world where the dire wolf, the legendary beast of the Ice Age, roams the earth once more. Yes, you heard that right. Thanks to cutting-edge science, extinct species like the dire wolf may no longer be confined to history books. But how far have we really come in resurrecting these ancient predators? In today's video, we'll dive into the controversial world of de-extinction, exploring whether the birth of the new dire wolves is a true scientific breakthrough or just a high-tech illusion. Stay tuned as we unravel the mysteries of DNA, genetics, and ethics behind this fascinating quest to bring the past back to life. Good morning, friends. Welcome to my channel, Career Capsule. Dire wolf, scientific breakthrough or high-tech illusion. The revival of extinct animals is no longer confined to science fiction. Colossal Biosciences, a company at the forefront of de-extinction, has set its sights on resurrecting the famous dire wolf, a species extinct for 13,000 years. They announced the birth of 3 genetically engineered dire wolves, Romulus, Ramos, and Khaleesi. These pups are thriving in a secret American facility, marking a remarkable achievement for the company. However, there is significant controversy surrounding this breakthrough. While the public is captivated by the idea of bringing back such an iconic species, scientists question whether these animals are truly dire wolves or just hybrids engineered to resemble the extinct creature. The heart of the debate lies in the methods used to recreate the dire wolf, which vanished so long ago. Can ancient fragmented DNA truly be reconstructed into a full genome? This is the central challenge of the project, with many experts urging caution and calling for rigorous testing to understand exactly what type of wolf these animals represent. The initial excitement surrounding Romulus, Ramos, and Khaleesi is tempered by skepticism about the authenticity of their genetic makeup. Dire wolves, ancient beasts with a lone evolutionary path. The dire wolf, Canis dirus, was a distinct species with a lineage separate from that of modern wolves. For years, scientists believed that the dire wolf was simply a larger version of the gray wolf, but a 2021 study revealed a surprising truth. The dire wolf diverged from other canids a staggering 57 million years ago, forming a unique evolutionary path. In 1918, scientist John Merriam proposed that the dire wolf should be classified separately, but his theory was dismissed at the time. Recent findings, however, have vindicated his ideas, revealing the dire wolf as a unique species with no direct evolutionary connection to modern wolves. One of the most intriguing aspects of the dire wolf's existence is that it coexisted with gray wolves and coyotes for thousands of years in North America. Yet no interbreeding ever occurred. This separation of genetic lines is crucial in understanding their eventual extinction. While the dire wolf thrived for millennia, it vanished around 13,000 years ago, likely due to the extinction of its primary food source, megafauna like mammoths and giant bison. Unlike gray wolves, which could adapt by interbreeding with other canids, the dire wolf lacked this flexibility and ultimately disappeared. The quagga comeback, recreating the past. Through selective breeding, de-extinction is not a new concept, and one of the most prominent examples is the quagga project. The quagga, a unique zebra subspecies, went extinct in 1883, but scientists have been using selective breeding to recreate the animal. The process, known as backbreeding, involves identifying modern animals with traits similar to the extinct species and selectively breeding them to amplify those traits over successive generations. This method has shown success with the quagga, as zebras with fewer stripes, particularly on their rear ends, were chosen for breeding. Over 20 years, the project has successfully bred zebras that closely resemble the quagga. However, this raises a crucial question. Does visual resemblance alone constitute true de-extinction? While the quagga-like zebras look similar to their extinct ancestors, they may not possess all the traits of the original quagga. The debate remains: is it enough for an animal to look like its extinct counterpart, or does it need to have the same genetic and biological characteristics to be considered a true revival? Inside De-Extinction: how scientists bring back lost species. De-extinction is an exciting but challenging field that involves multiple methods for resurrecting extinct species. The three primary approaches are backbreeding, cloning, and genetic engineering. Backbreeding, as demonstrated by the Quagga Project, involves selectively breeding modern relatives to bring back lost traits. While promising, this method doesn't guarantee a precise genetic match to the original species. Cloning, on the other hand, aims for an exact genetic copy by transferring the DNA from an extinct animal into the egg cell of a closely related species. Although this method holds the potential for more accurate results, it is fraught with difficulties, as demonstrated by the failed attempts to clone the Bocardo goat and other species. The most promising approach, according to many researchers, is genetic engineering. This method involves using advanced tools like CRISPR-Cas9 to edit the DNA of a living species, introducing genes from the extinct animal. While this technique is still in its infancy, it offers the possibility of a more controlled and precise recreation of extinct animals. The challenges remain substantial, but as technology advances, genetic engineering could become the key to unlocking the potential of de-extinction. DNA editing and the dream of de-extinction. The core of de-extinction lies in DNA editing, which involves altering the genetic material of a living species to recreate the traits of an extinct species. To do this, scientists extract fragmented DNA from fossils, often broken and incomplete, and use sophisticated techniques to piece it together. Once a workable sequence is obtained, the DNA is inserted into the cells of a closely related species. The edited cells are then cultivated and, in some cases, implanted into surrogate animals to create offspring with the characteristics of the extinct species. The dream of de-extinction has captured the imagination of scientists and the public alike, but the process is not without its challenges. The most significant hurdle is obtaining high-quality DNA from ancient fossils. As DNA degrades over time, extracting usable genetic material becomes increasingly difficult. Additionally, editing millions of DNA sequences in a precise and controlled manner is an immense scientific challenge. Despite these obstacles, researchers remain optimistic that advances in technology will eventually make de-extinction a reality. Cracking the code: how scientists reconstructed dire wolf. DNA. Reconstructing the DNA of the dire wolf is a monumental task that has captivated the scientific community. In 2023, researchers at Colossal Biosciences embarked on the project of bringing back the dire wolf using genetic data extracted from ancient fossils. They obtained crucial DNA samples from a 13,000-year-old tooth fragment and a 70,000-year-old skull, providing a wealth of genetic information. By comparing the dire wolf's DNA to that of the modern gray wolf, researchers have identified the genetic similarities and differences that will allow them to recreate the traits of the extinct species. The process of recreating the dire wolf involves using CRISPR technology to modify the DNA of gray wolves. This approach takes advantage of the close genetic relationship between the two species, making it easier to introduce specific genes that will recreate the dire wolf's physical characteristics. The birth of Romulus and Remus, the first genetically edited dire wolves, marks a significant milestone in the project, demonstrating the potential of gene editing in de-extinction. Engineering extinction in reverse. The birth of Romulus and Remus. The birth of Romulus and Remus, the world's first genetically edited dire wolves, represents a major breakthrough in the field of genetic engineering. Using CRISPR technology, researchers altered 14 genes in gray wolves to modify traits such as size, head shape, and fur density. These gene-edited cells were then used to create embryos, which were implanted into surrogate dogs. Despite the challenges, 2 pups were successfully born, marking the first step toward bringing back this extinct predator. The process behind their creation was complex and required precise genetic manipulation. Scientists used CRISPR-Cas9, a tool that allows for targeted edits to DNA, to achieve the desired traits in the dire wolf clones. Although the pups are genetically identical, their personalities differ, illustrating the role that environmental factors play in shaping behavior. This underscores the idea that cloning does not guarantee identical outcomes in terms of behavior and personality. Cloned but not cloned alike. Nature, nurture, and the new dire wolves. While Romulus and Remus share identical DNA, they exhibit different personalities, highlighting the role of nature versus nurture. This challenges the common assumption that clones are exact replicas, both physically and behaviorally. Just like identical human twins, who may share the same DNA but develop different personalities, the dire wolf clones have distinct traits shaped by their environment and experiences. This serves as a reminder that cloning is not a straightforward process, and even genetically identical animals can exhibit unique behaviors. As the dire wolves grow, their environment will continue to influence their development. This raises questions about the potential for cloning to truly recreate an extinct species. While the genetic makeup may be accurate, the lack of a natural upbringing, such as being taught to hunt by their parents, could affect the clone's ability to thrive in the wild. The project's limitations, including the inability to allow the wolves to breed, raise ethical concerns about the future of these animals. The ethics of de-extinction. Just because we can, should we? The question of whether we should bring back extinct species like the dire wolf is a deeply ethical one. While the scientific achievement is impressive, the core question remains: why are we doing this? If the goal is to restore an animal's ecological role, the project may hold promise. However, if it's simply to create living exhibits or novelty animals, the ethical implications are more troubling. De-extinction should not be about spectacle, but about understanding the ecological roles that these creatures once played. The dire wolf's potential reintroduction into the wild raises questions about the ecological impact. As an apex predator, it could help restore balance to ecosystems that have suffered from the loss of such predators. However, the artificial nature of their upbringing and the lack of natural socialization could hinder their ability to thrive in the wild. The ethical debate centers on whether we should use our technological advances to resurrect species or focus on protecting the species that still exist today. As we've seen today, the idea of resurrecting the dire wolf is both thrilling and terrifying. The science is advancing, but with it come deep ethical and ecological questions. Are we ready to face the consequences of bringing back the past? Let us know your thoughts in the comments below. Do you think de-extinction is the future, or just a step too far? If you enjoyed this dive into the world of ancient science, don't forget to like, share, and subscribe for more videos like this. Until next time, keep questioning, keep exploring, and keep learning. Jai Hind.